And we are ready to go now. So the topic is manipulating variable lists and prioritizing your results. The way I see this, I wanted mostly to talk about different approaches, so you will get ideas how to handle complex results when you work with complex data sets, and type of data is complex, and this complexity is only increasing and will only be increasing. So the right assumption is that we need to jump into it and to understand and to create a strategy for ourselves and to keep learning things, as it will not be getting simpler. Look, we look at the new complexity which is introduced by single cell data, multi-omics signatures. Here we will be talking about multi-gene signatures, but then next levels will be layers, multi-omics signatures, single cell. It's the way it is right now is easier to get started for those of you new to this area. So let me just jump. Okay. So I assume we have all kinds of people here today, our customers, and also new to Qlucore people, and those who are evaluating Qlucore right now. So I will just give a little overview about the tool very briefly, and about the data types which you can import. So the main strengths which Qlucore will bring you, apart from the algorithms and outstanding speed, is that it will help you. It will be enabler and pattern seeker and prioritizer. It will aid your brain and your mental manipulation with hypothesis and discovery. You will get very good feel and understanding for your data using 3D interactive plots, flexible ways to work with lists, and you will be able to zoom in and out really fast on subsets, on results, to overview of the complete data set. And of course, the most fun part maybe is beautiful, stunning visuals for presentations, record keeping for publications. And with the speed, I mentioned that, for example, if you look at just PCA plot created, it will be generated on the regular laptop, like I'm working on the MacBook. It will be generated 77 times faster than the same plot will be created in R, which is a big benefit for you working on your regular computers. And also it's a huge benefit for mental processing of the information, as long interruptions between steps and pictures will be slowing down and making the way you think about your work less efficient. And this is the message mostly for biologists, physicians, people who are most often these days are distanced from making results and decisions about data analysis. You are positioned best to make these decisions. Really, there is nobody else who knows your projects and experiments as you do. So this is a good time in the scientific world to jump in and to start being in charge of your data and analysis. What kinds of data can be imported into Qlucore? You can work with gene expression data, both arrays and RNA-seq. We support single-cell data as well, proteomics, metabolomics, lipidomics, protein array, DNA methylation arrays, flow cytometry, qPCR data. So what's coming next in March probably is support for ChIP-seq and ATAC-seq. Yeah. If you have some other questions about your particular data, please type them in the chat. And now I want to show you an overview of what I would like to show you today. As we look at the big data sets and complex data sets, there is no one best variable subset. There is no one best resultant list. You will have to make a decision. You will look at the range of results, a range of subsets. And we give you some aid and guidance in Qlucore to make you feel confident and evaluate them. before you choose what you will decide as your final result. And of course, iterative analysis, you can always go back and reanalyze and compare your results. Maybe you will be merging different experiments together into a bigger data set. So this all is possible. So ways how we can look at, I will be working, by the way, with gene expression data sets. So I will be using gene terminology. It applies to any, like it can be proteomics, metabolomics, lipidomics, any variable, any variables you're working with, just using genes here. So we will look at, we can look at the layer of highly variable genes. We will see which, those are the genes which will be introducing the strongest signal in your data, creating patterns. Another take is, of course, looking at statistically significant genes. And then we can look at number of statistics here. I will set up a ANOVA multi-group comparison and two-group comparisons, and we will use P and false discovery rate Q, Benjamin Hodgberg, to filter, to sort and filter results. We can look at enriched pathways when you have when you use GSCA, for example. You can zoom in on an individual pathway, having the list of genes which comprise that pathway. I will do it too. A single pathway of interest. So you can see how this pathway behaves, and you can see which genes of this pathway are statistically significant in your groups. We can look at existent, like published condition signature, what I will do, and you can see if this signature works in your own results, and vice versa. You can create a signature using your data set for a condition and then finding some other published or somebody else's data set of similar conditions to see if it works there. You can zoom in on the list of therapeutic targets. That's pretty much like a gene list, which you create, and then you can, so it's one of the variations of the variable or gene lists manipulation. But in the end of the day, this the decisions and cutoffs and approach how you combine different cutoffs, different layers, will depend on your research goals. So, for example, if you're in a discovery mode or confirmatory analysis, so in general, discovery mode, you won't be nearly as stringent as in, for example, guidance for diagnostics. If you work with patient data, then your tolerance for false discoveries will be very low. While in discovery, if you tolerate, let's say 1% or even 5% of false discoveries max, what happens, you will lose real discoveries. You will never get to see them. So that's a balance. It depends on your what you do with the data. And also your assumptions. It's really good to know, and you will be learning this as you go. It's not always that you know all this information. For example, assumptions about the effect size of what you are studying. Let's say you are looking at the effect of some compound on the cell culture. And then you may have already an idea what scale of the changes will be happening. Will it trigger really major perturbations, or will it be a mild regulation effect or very minor? So if you know this, you can comfortably check, of course, but you can be more comfortable filtering on fold change. If you expect very low scale changes, then filtering on fold change severely, you will lose real findings. So, okay, let's switch to the program. And I have opened acute lymphoblastic leukemia dataset, which is supplied with the software installer right here in help example files. And I collapsed I log transformed this data set right here in Qlucore and transform, and I collapsed identifier on gene symbol. So right now we have 13,266 unique genes right here in this list. And this is a sample PCA. Each dot is a sample. 132 samples are right here. So I will create a synchronized variable PCA, and we will look at both genes and samples. So left is plotting all 13,266 genes, and on the right we have 132 patients. So what I will, let's look, I wanted to show you filtering on variance. Thank you for the note. I realized that I have to give WebEx permission to share. I will just share the screen. Yeah, thank you for the note. So right now, you should all hopefully see my biplot. I will color this sample by samples. This data set is looking at leukemia subtypes. So I just colored my patients by the subtype diagnosed cytogenetically. And on the left, we are looking at our genes. And genes, I want to color too. Let's color them by standard deviation, by variance, and this color scale is maybe not. Okay, this one is better. Okay. So what I want to do, I will bring here our statistics and variance management window. And I want to start filtering by variance. Right now in real time, we will see all the different variable gene subsets based on filtering low variance layer by layer. So, and if you can pay attention to this lower left corner, you will see that number of genes will be decreasing as I'm sliding the slider to the right. So for example, right now you can see this very nice, more clear clustering of our patients by subtype. And on the left, we see fewer genes, 1,120. So this is the clustering of patients, which takes place with this very 1,120 most variable genes. So if you keep, I will do it, I will overfilter, of course, just for the demo, 49 genes. If we keep these 49 genes, and the list of these genes, by the way, is right here in variable space, so you can always keep track of what genes, especially if the list is not too huge. And that's, you see respective changed instantly patient input clustering of samples. So if I will filter to, okay, this is filtering, really extreme filtering, 15 most variable genes, but they're all red because the variance of those genes across the data set is very high. And you see that the pattern in the sample clustering keep changing with changing the subset of genes. Like this 553 gene input really separates one leukemia subtype, but the rest are clumped together, even though you can see the difference. It's not still a random cloud. So that's the idea. And you can save, let's say if I am interested, what I tried to optimize variance, there is a way to control to overfilter. If you overfilter variables, we deprive data set of the information content, which is just a warning sign. You need to be aware, you need to have a reason to, so let's say I want to save this subset, 289 genes. I will make a copy here in variable list, and this is my 289 gene subset, most variables. So you can write a note to yourself here. So that's the idea. And why? What's this variance filtering doing? It's a noise, controlling the noise reduction. If your data set is, and omics data sets are noisy, some are more, some are less. especially for proteomics, metabolomics. This step will be crucial. Single cell will have a lot more noise as well. So removing low variance will show you clustering, of course, this example clustering, and also it will increase potentially statistical power, statistical resolution power. Okay, and let's now, it brings us nicely to statistically significant genes, filtering by statistical parameters. So what I will do, I will clear this variance cutoff, and we will go to statistics. And let's set up two-group comparison, comparing TLL subtype with the other subtypes, and I will look for both up and down regulated genes. So here you will see one slider with two windows. One will give you control over P cutoff. Let's say if I would like to go with conventional P 0.05, I just click enter, and the result is instantly available. 7,242 genes, lower left corner, and also right here are kept with this P cutoff. And corresponding Benjamin-Hochberg Q is calculated to the right. You will see it's right here, 0.09. So about 10% of false discoveries max. And in the list of genes right here in our results, you can add both P and Q values. And also here, with the two-group comparison, you can calculate a fold change. It's so another way, before we go to the fold change, very often you will want to control Q and not P. So if you have a hard stop on false discoveries limit, let's say if this were used to guide diagnostics, probably this would be the highest acceptable false discovery rate, 5%, which still gives us a lot of variables. This TLL subset is very distinct from the other subsets. It's not always like this. So maybe we can go to 1%. And even with 1% of false discoveries max, we end up looking at 4,333 genes. So I will save this as my intermediate result. We'll just call it TLL versus the rest. Q or FDR 1%, right? And so that's, you will see nice by plot our genes and our samples. So now fold change, because it's a two-group comparison, you can enforce fold change as well. And you can, even here with P and Q, you can use the slider to observe the behavior in real time with different stringencies. And both P and Q are recalculated instantly. So let's now start with this 5P 0.05, and then in the fold change, what I would like to do, I would like to start changing fold change right now. And here is the slider on the bottom, and you will see that here is a good way to look at the whole range. So we are, it's log 2 fold change calculation, not ratio, and you can see the range by using the slider. Or if you have a specific fold change in mind, you will enter it right here, press enter, and here is my result, 35 genes from my data set will pass P value 0.05 and fold change 1.5. This is the result I would say for a fold change. Okay, so now let's look at some specific pathways. I wonder if I will have time for GSEA. Let's see if we'll time. I will quickly run GSEA in the end. But right now, I want to explore P53 pathway. And what I will do, I will clear of course, here you would go and create a beautiful heat map, and here I will do that fast. Nice to see synchronized changes on the heat map and PCA. So we'll go with leukemias as ordering by subtypes and variables by clustering, and I will color my samples by my annotation, my main annotation, and my variables I will color by, let's say, by fold change. And here is what we get. Okay, now we see complete input for both samples and variables, and you see they present the same information in two different ways. The most distinct on the heat map subtype is the orange one, which is TLL, and here it is. You can see just the same. We were comparing TLL with the rest, yeah, that's it. So let's now go and find how P53 pathway behaves in this data set. I have it saved as a text file, no header, nothing, just one gene per line. And let me import it, file, import, variable list. And it's right here, we'll mark collection of MSigDB. It's brought into your variable management space, which makes it easy to work with. So what I would like to do, I want to limit the view of my data set to this P53 pathway. So now we are looking at 193 genes. Here they are. And you can see that P53 pathway will not separate our subtypes. So I will just focus right now on the heat map. You see 200 in the first column and 193. It means the set which I imported has 200 genes, but we have only 193 matches in my data set. So some of them were filtered maybe during the noise reduction for this data set. In addition, so this is just the behavior of the pathway, and you can see the genes which comprise this pathway right here, and you do see the patterns. And maybe, well, it's hard to tell if the TLL is more distinct. I don't think it is visually. But let's apply also statistical. You can layer different cutoffs here on the top of the P53. Let's see if for TLL comparison of TLL with the rest, let's see which genes will pass my P0.05. So half of them, 108, remain after P0.05. And then, of course, you will go and play with the Q1.5 as we did earlier. And none of those genes are variable to that degree. So, but then you can find out what is this. Okay, here you go, 1.2 or so variance. Okay, this is the highest, 1.26, most variable gene from P53 passing P0.05. Okay, that's just an idea. And another way, if you have one of those genes in mind to stratify your samples by abundance of the gene, by abundance of the gene expression, we can do that. Let's pick one of them right here. And I didn't show you. Sorry, you can sort your results in the table, clicking on each of the statistics. Now I sorted by p-value, lowest, the smallest values first. So this is my top candidate. If I'm guiding myself by p-value. If I'm guiding myself by the q value, let's see, it's still the same gene, PLX and V2. If I would like to look at the highest fold change, then which one? These are missing genes which are not present, not passing this cutoffs. So this is the highest, my top candidate based on the fold change. So that's how you will look. You can look at this results in different ways. But what I would like to do to show you right now, I want to sort this heat map by the expression of one gene. If I had one gene in mind, which would guide my stratifying these samples, I would go to View, Order Samples by the value of the variable. So in this case, it's a gene symbol, and I will type the name of this, let's say AGA. Actually, this is not very intelligent. It's just a blind choice. I'm not sure it makes any sense, but I just wanted to show you. And here is the gene. Let me zoom in so you can see better. I think it's, yeah. You can see it, we have this beautiful gradient of expression. Yeah. And based on this, we have high expression right here, and lowest expression is on the right. And so here is where you can make decisions and create new sample annotation. Highly expressed AGA, lowest expression, and then you can make as many groups as it makes sense for you. And we will create new sample annotation right here, and you can set up different kinds of analysis. So it's mostly for the discovery step. So now I want to show you how I will bring, that's my big heat map. I will import published signature. I will bring the list, different list of genes differentiating this leukemia subtypes. And this one. This is the list of 59 genes, and you can see that I have 46 present in this data set. So if I zoom in on this list, and this is my sample PCA, you can see that it does separate. The question is, let me check my, okay, oops. Yeah, I need to clear my statistical cutoffs. So what we see, and TLL, this orange group is separated nicely, and you can see also some pink, and so it definitely separates our subtypes. And let's apply statistical filter, stringency of your choice. Let's say if I apply P0.05. So then only 29 of these genes of 46 will pass this cutoff. So then you would say, yeah. So there is a discrepancy how this signature works for me and how it was published. Then you would go back and you would need to know exactly how that research group who published this signature, what cutoffs they used, and of course it's different data set. And the other way around, if I build a signature right here in my data set, let's say this one, this is my signature for TLL, and then I can use public data set and bounce this signature and see what will happen in that data set if I use my signature. Will it be still separating? Now I see nice separation of orange group TLL leukemia with the rest of subtypes, and that's what I expect. And sometimes signatures often, I think, will be directional. In my case, I used both up and down regulated genes to build the signature, and the same way published signature, just the genes which separate groups. But you may have a signature which will be looking for only upregulated, pointing to some genes up or down regulated in the specific condition. So let me look at the time. Yep, so we are just over 30 minutes. And one thing I wanted to do is to show you how to set up a pathway analysis, pulling this enriched pathways as a new layer after your statistical and variance. But this P50 is one of the results from gene set enrichment. So the difference is when you run GSEA with your data, you will often have 50, 100, 150 sets enriched to different degrees. And then you will be looking at functional meaning and how it relates to your project to pick. And that's something where only the project owner, the researcher involved in the project, can make those decisions intelligently, as you need to know a lot of background. Let me stop sharing this screen for a second. I want to see the questions. So yeah, now it's a good time to send your questions, and I will be glad to answer or to demo. If something was not clear, I can repeat some steps. Yep, let me, I am going back to the screen sharing of the Qlucore screen. That's what you should see now. And let me show you. Yes, so that's about the GSCA. I will remove all the cutoffs, all the input filters, as GSA requires the data set as an input. Here is the button to start GSCA. Oops, okay, my bad. I see that P cutoff is still not clear, but I will to save us some time, I will limit variance. Okay, let's say this subset. This is normally what you wouldn't be doing all the time. So I'm using hallmark collection, downloaded from the Broad's website. And here I to indicate what tests I'm using. I'm using two-group comparison, both up and down on leukemia subtype TLL versus everything else. And just press run, and let me see, symbols and no matches. Probably I filtered. This is too small subset. Let me see. I'm working on gene symbols. Let me just have more genes. Okay, let's use this in. Waiting. Oops. Sorry, it's freezing a little bit. Screen share can be a little tricky with this intense computing we're doing. So I'm using the hallmark set to group comparison. And while we're doing how to mark, so another question, how to mark and color label variables on the heat map. Yes, there are many choices. There are many choices, and I will show that quickly after this. You can pick your label, you can change the label size, you can color them in different ways, and you can also mark, if you would like sometimes to highlight gene list or one gene. So now we have results of enrichment for this setup, and this is what we get. That's what I meant, that here you will do more. Exactly, we got 50 hits. And the gene set name is gene set name. So hallmark is the collection, and then cholesterol homeostasis. Now, for example, size, how many genes are in the set, and how many matches do we have in our data set. And then you have enrichment score, normalized enrichment score, PQ, and description linked to the set. I will be guiding myself here with NES. Normalized enrichment score, the higher the better. And then my top candidate is cholesterol homeostasis, then androgen response, and mTOR signaling. Just remember, I used a subset. This is not a complete data set. Then DNA repair and checkpoints, so more cancer-specific pathways. Let's choose like one or two. You can bring all of them back if you like. I'm choosing only two. Click list, and then they appear in my variable management space. And let's say I will limit the view to mTorque signaling, and let me show this thing with the heat map. So select heat map, and then in the view tab, you can, I will automatically size it so we see all of the input samples and variables in the view. I will color my samples by annotation subtype. This is my main annotation, and then you can add all of them, or specifically the ones which will make sense to use, so visually not to clutter. Coloring variables, genes here, so you have a whole array of options by list, and I did not color our lists, but you can give them colors. Our GSCA list, hallmark mTorque, this one a MIC targets. So you can see that now they start populating. So mTorque is turquoise, and red ones are coming from MIC targets. This is, so that's coloring by list. And color by variance. So the red ones, deep red, are the most variable gene, most highly variable genes. You can use the Q value also. You can apply fold change coloring, and you can color by the normalized gene expression in a specific group, choosing right here. So for example, for leukemia type, TLL. So we'll see the expression in TLL, average expression, orange group, just in average form. Individual samples, patients, and this is averaged. And labels, now we have probe sets. I will switch to gene symbols. We can see them because of the size, it's too small. And now I'm zooming in, you can see gene symbols are populated, and yeah, you can apply hierarchical clustering of the samples. So let me go back for a moment so I can see the chat. I hope that's clear. Yep, and another question about the variance. Let's go back quickly. This, I think, is the last we can do today. Is variance. I will switch to PCA for faster processing, and will bring my variance control in statistics variance. So variance is looking at the variation of each variable in the data set in regarding to the maximal variation of the maximal variance in the data set. So this is kind of noise reduction approach. If we treat small variance, low variance as a noise, which is true for gene expression, metabolomics, proteomics, lipidomics, for sure. If a small variance is a noise, then what we're doing here, and if I have again one variable genes, one variable PCA and one sample PCA, these are my variables, genes, and samples. What I'm doing here right now is I'm removing noise, and I'm focused only on one pathway. It's okay. It's not a complete data set. Normally you do it on the whole data set though, because that's where you clean the noise. When you have too many genes which have low variance, it will be kind of blurring the patterns which are emerging. Like, look at the sample PCA right now. It's obviously not random. You can tell that this sample type distribution is not random, but you don't see nice clusters or any really clear clusters of subtypes. Now, when I'm reducing variance, when I'm increasing the limit, right, filtering out low variance genes, you can see now we look only at 111 genes, most variable genes in the data set. And you see what happens with the samples. It's because we remove the low variance noise, which helps us see the pattern. This is a stronger signal in the data set. And as a result, if you set up, not always, but often enough, if you set up statistical tests, especially if your effect size is not that big, you set up statistical tests and you don't find enough results, so you feel like your false discovery rate is too high. You find you expected a lot more genes after the statistics. Then by removing low variance, small variable genes using this variance filtering, you often end up improving your statistical power. So you will have better statistic and more genes which will pass the cutoff. Especially when you work with machine learning, when you build predictive models for a condition, that's where noise becomes, noise reduction is important step always for every analysis. But when you want to build predictive modeling used potentially in guidance for diagnostics and some medical settings, that's where it becomes a lot more critical to know your noise in the data. This is just one type of the noise. Another type of noise will be batch. If you have batches or samples processed in different locations, the same, yeah, it's a batch too. Yeah, you would address those as well. But this is one type of noise, is low variance. And in metagenomics, it can be the other way around. The highest variance, if we're metagenomics, some data set, then maybe the highest noise signal would be your noise, because then you would think it's potentially contamination of the sample. And there you would be removing the highest, highly variables and not. But in gene expression, protein data, metabolomics, this approach with removing low variance works well. Yes, I hope it helps. And we are at the end of our time. Let me, I shared the link in the chat before if you're looking to update your installation with the newer version, or if you're looking for the trial, it's the same link, you can use it. And thank you so much for joining today. Wishing you happy holidays. And many of you will probably use this extra time to work with the data. We are here to help if you need any assistance. Thank you so much. Bye. Bye. Bye.